Hello everyone, so this is the GCSE Science Trilogy Biology Paper 2 walkthrough, and this was the paper that was sat by pupils in June 2019. I'm going to go through how I would have tackled this paper. I've put links to the blank paper and Markscheme in the description, along with revision video links, question by question if you need to go back and revise that topic. So, number one then. Conditions inside the human body are controlled. What is the control of conditions inside the body called? Well, excretion, that is the removal of waste from the body, so it's not that. Fertilization is when you've got a sperm and egg fusing, so it's not that. Osmosis is the movement of water, so it's not that. So therefore it is homeostasis. The definition of homeostasis is how our body maintains a constant internal environment, and that is important so our body works at the best conditions it can do. So question 1.2 then, what are the two ways information is sent to control body conditions? So how does information basically get round our body? The two ways that it happens is by hormones, which are chemical messengers, and our nervous system by using nerve impulses. Red blood cells, their only function is to carry oxygen, so it's not that. Muscles just contract, they don't send information. And antigens, these are the little molecules on the surface of a cell membrane. That's to do with our immune system, so it's not that. One condition that the body needs to be controlled is the level of water. Give another condition that needs to be controlled. So the two other key homeostasis conditions that our body regulates is our temperature. Now body temperature 37 degrees Celsius is important because we need enzymes working at the correct level, and also blood glucose. So just by saying blood glucose or temperature, that would get you the mark. Figure 1 shows the volumes of water taken in and lost by a person. The volume of water taken in on a hot day has not been plotted on the bar graph. The person lost 1,400 centimetre cubed of water on a cold day. How much extra did they lose on the hot day? So we need to work out how much water has been lost on the hot day, and then compare it to the amount of water lost on the cold day, and therefore we can then see how much extra was lost when it was hot. So we can see urine is 600, sweat is 1,000, and then breathing is 400. So that gives us a total amount of 2,000. Now if we look at the cold day was 1,400, and the hot day we've just worked out was a loss of 2,000. So if we now subtract it, that gives us a difference, 1,400, so that gives us a difference of 600 centimetre cubed. Question 1.5 then. Explain why the volume of water lost on a hot day is higher than on a cold day. So why might we lose more water when it is hot compared to when it is cold? Well, hopefully we realise if it's really hot, what does our body do? We sweat when it is hot. And the reason we do that is because the sweat cools the body when it is hot. So 1 mark for identifying that it's sweat, and then 1 mark for identifying what sweat does. A boy drank 750 centimetre cubed of water, and his total intake of water was 3,000. Calculate the percentage of the boy's intake that the 750 centimetre cubed represents. So we're just doing a percentage. Now when you do a percentage, it's pretty much always the smaller number over the bigger number times by 100. So 750 out of 3,000 divided by 3,000, times that by 100, put that on your calculator, and it gives you 25%. Okay? Question 2. Some students estimated the population of daisy plants in a field. This is the method they used. They placed a quadrat randomly on the field, they count and record the number of daisy plants in the quadrat, and they repeat steps 1 and 2 another 4 times. How could the students have made sure the quadrats were placed randomly? Okay, now this is one of the big errors in this practical, that people just go outside and start chucking quadrats. That is not an accurate way of doing it. The best way to do it is to get a diagram of your field. You then divide it up into 1 meter grids. You assign each of them a number, and then you would use a random number generator, which would then tell you where to place the quadrat. Okay, so that removes any bias from it. Okay, so could the students have made sure the quadrats are placed randomly? Well, you could use a random, there's only one mark, so just say random number generator. Question 2.2. Describe the piece of equipment called a quadrat. So you just need to say for 1 mark, what does a quadrat look like? Well, normally it's a 1 meter by 1 meter square grid. Now, most of the time it's a meter by a meter. However, you can get them at half meters, 0.5 by 0.5. Question 2.3 then. You've been given the results below. So you've got the quadrat number and you've got the number of daisy plants in the quadrat. It then asks you to calculate the mean value. Well, the mean is the same as the average, so you would add them all up, okay, which would give us 19, 23, 29, 39, 45. That gives us a total of 45, and it's divided by 5, so 45 divided by 5 is 9. The rectangle, the field is a rectangle, and it is 100 metres wide and 150 metres long. So you've got to calculate the area. So it's just the area of a rectangle, 100 by 150. Hopefully we know from maths that to do area is just base times height. So 100 by 150 is 15,000. The quadrat used by the students had an area of 1 metre squared. So they were using a 1 metre by 1 metre quadrat. Estimate the population of daisies in the field. Use your answers from 2.3 and 2.4. So this will allow if you made a mistake on the past 2 questions, you'll still get credit if you follow this step. Now, what you've worked out is that there is an average of 9 daisy plants per metre squared, and you've got 15,000 metre squares. So in order to estimate the population, you would just do 15,000 times by 9, okay, which gives you 135,000 daisy plants as an estimate. Question 2.6. More daisy plants grew in some parts of the field compared to other parts of the field. Give 2 biotic factors that may affect where daisy plants grew in the field. Now, a biotic factor is a living factor, or something that is alive that affects the number of daisies. We can't then say things like the amount of sunlight or the amount of water, because sunlight and water aren't living. They are abiotic factors. So some biotic factors you could say is, well, it could be any herbivores, the amount of herbivores about that would eat it, or other animals. Or it could be down to humans trampling them. If you go on a school field, you'll always see there's less daisies around the football pitch because they've been squashed by people playing football. You could also have humans mowing the lawn or the field. Okay, therefore there'd be less daisies. Question 2.7. The students noticed that the daisy plants growing near the building were smaller. Explain why smaller daisy plants grew near the building. So we've got to think, what is the building preventing for the daisy plants from, why is it preventing the daisy plants from growing? What is the building doing? Well, the building, think about what plants want. Plants want light. If they're smaller, it means they're not getting as much light because they're doing less photosynthesis. So you could say the building gives less light or blocks out light. So therefore, the daisies would do less photosynthesis. And therefore, you would then have potentially fewer or less sugar. So therefore, there's less food for the plant to grow. Question 3 then. Animals have adaptations to help them survive in the environment. These adaptations may be structural, behavioural or functional. Draw one line from each of the animal adaptation to the type of adaptation it is. So you've got the animal adaptation, you've got male palm cockatoos use sticks to beat hollow branches to attract females. You've got the harmless hornet moth has black and yellow stripes to look like a bee or wasp. And you've got sea spiders that have an automatic muscle contractions that move oxygen around their body. And we've got 3 different types. We've got structural, behavioural and functional. Now a structural adaptation is a specific feature. So what you can physically see, like claws on an animal. A behaviour, well, that's self-explanatory, is how an animal behaves or works with others. And a functional adaptation is what happens inside the human or inside the organism's body. So, male palm cockatoos use sticks to attract other females. Well, that is what they are actually doing and how they're behaving. So it would be a behavioural adaptation. The harmless hornet moth has black and yellow stripes, where it's got physical features then that you can see. So it would be a structural adaptation. And therefore the functional one must be the spiders, and that is because they have automatic muscle contractions which happen inside the body. Plants also have adaptations. Orchid plants have adaptations which allow them to be one of the most successful plant groups. Orchids rely on insects for pollination. Which 2 features help orchids survive? So we've been told they use insects for pollination. So that is going to be our key hint in this question. Well, if you've got an insect, okay, they are going to be attracted to bright coloured flowers. And an orchid would want to pollinate, so having large quantities of pollen would also be useful. So I'm immediately inclined towards those 2 answers, but let's just double check. Right, no scent. Well, no scent wouldn't be an advantage because you'd want to attract the insects with a nice scent. The shape of the leaf wouldn't have any particular impact on it, and having small leaves, well, that wouldn't help it survive because you'd want a bigger leaf for photosynthesis. So it would be the top 2 that help insects pollinate. Okay, so question 3.3 then. Many orchid species grow in tropical rainforest ecosystems. What name describes the variety of all the different species found in an ecosystem? Tick 1 box. Well, the habitat, that is the area in which they live, so that's not describing all the different species, so it's not that. Feeding relationships is not that. Evolution is about how animals change over time, so it's not that. And it is biodiversity. Biodiversity is the amount of species living in a specific area. So a rainforest would have high levels of biodiversity because there's loads of different animals living in it. A school field would have very low biodiversity because it's just grass. Question 3.4 then. Some species of orchid may become extinct because of deforestation. Give one reason why tropical rainforests are being cut down. So, why are we cutting down the rainforests? Well, it could be because we want for wood for materials, or building, okay, as well as paper or fuel. And you could also say just for the space to build and also to farm cattle. Question 3.5. Give one factor that might cause the species of orchid to become extinct, but you can't refer to deforestation in your answer. So what might be another reason why the orchid becomes extinct? Well, these are your general factors of what causes extinction. You could have a new disease or pathogen that could wipe it out. You could have new herbivores that come along and eat it. You could say climate change, that is reducing biodiversity, or you could say a new competitor. So maybe it's a new flower that comes along, that evolves, that has even more pollen, or that even has a nicer scent. Question 3.6 then. Scientists have analysed the entire genetic material of one species of orchid. What is the genetic material made from? Okay, so what actually is genetic material? Well, we should know it is DNA. And then 3.7, what is the name for the entire genetic material of an organism? Well, it is a genome. That is very specific knowledge, but a genome is one copy of an organism's DNA. Question 4 then. A cat breeder noticed that 4 kittens from one Siamese cat mother had a new blue colour at the end of their tails. What has caused the new colour to appear? So this question is about inheritance and genetics. Tick one of the boxes. Why has this blue colour now appeared? Well, fertilisation is when a sperm and egg fuse together, so it's not that. Mitosis is making new identical cells, so it's not that. So it must be a mutation. A mutation is a change in the DNA. Sometimes it leads to a new feature, sometimes, or actually most of the time, it doesn't do anything. The cat breeder wants to use selective breeding so that all new kittens have blue tail tips. Describe a process of selective breeding the cat breeder would use. So we need to say what selective breeding is. So selective breeding is pretty much about managing the sex and making sure that those animals with the desired characteristics, they are the only ones that breed, so therefore you are more likely to get that feature. So what you would do to do selective breeding for in this cat situation, you would choose the cats with the blue tail. You would breed these cats together. Okay, and then you would look at their offspring and you would then breed the offspring which have the desired features together. So you continually do it, you choose 2, they breed, and then you look at their offspring. Well, those 2 offspring have the desired features, so I'm going to keep breeding them. And you do this over many generations until all cats have blue tails. Question 4.3. Suggest one reason why the cat breeder wants to have all new kittens with the blue tail tips. Well, why do people do this sort of breeding? You could also link it think about pugs as well and the cross competition. Why do people do selective breeding? It's for money, because it would be rare. These cats would be rare or expensive. Okay, or you could say to make more profit. Okay, obviously there are ethical issues with it, especially with things like cross competition and the health effects that the offspring then have, but this is why selective breeders do this process. Question 4.4 then. Siamese cats can suffer from heart defects. Why might there be more Siamese cats with heart defects amongst the kittens with the blue tail tips? Well, they're not clones because there's still a bit of variation in them. They are not just formed from mitosis because it would be meiosis, because you'd still have sperm and egg fusing together. Just because they're produced by sexual reproduction wouldn't necessarily increase their heart defects, but it's because they're produced by inbreeding. If you've got constant inbreeding happening over several generations, it can cause health effects. With each pregnancy, the cat breeder expected that 50% of the kittens would be male and 50% would be female. The sex chromosomes in cats are inherited in the same way as humans. The sex chromosomes are X and Y. Give the combination of sex chromosomes present in a male cat and a female cat. Well, it doesn't matter that we're talking about cats, it's the same as humans. So a male cat would have the XY combination and a female cat is XX. So remember, it's our 23rd chromosome that determines our sex. You can either inherit a Y chromosome or an X chromosome from the sperm, and it's the sperm that decides whether you're a male or female. If that sperm has a Y chromosome that gets into the egg, you'll be male. If it's one that has an X chromosome, you'll be female. Question 4.6 then. The cat breeder expected 50% male kittens and 50% female kittens. Complete the Punnett square in figure 3 to show why. So what we do then, we know our genotypes. For the female is XX and the male is XY. So we separate out our genes into the different boxes, and then the boxes next to it, we join them together, and then we can demonstrate that we've got 50% female and then 50% male. Question 4.7. In the first pregnancy, there was 1 male kitten and 3 female kittens. Give the reason why there were not 2 kittens of each sex. Well, this is just because that wherever you end up, you could end up, if the offspring could end up in any of these boxes. Fertilization is a random process. Okay, so it just so happens, for instance, that 3 of the fertilizations happened and they got XX, and only 1 was XY. Question 5 then. Figure 4 shows a food chain in a garden. Which term describes the spider in this food chain? Well, on a food chain, it always starts with a plant, and we call that the producer, because they are producing the food. You then have the primary consumer, then the secondary consumer, and then the tertiary consumer. So what would the spider be? It is our secondary consumer. So the primary eats the producer, the secondary eats the primary consumer, and the tertiary eats the secondary consumer. Many of the spiders in the garden died. What is likely to happen to the number of black flies in the garden? Well, if all the spiders get wiped out, what would happen to the amount of black flies? Well, their prey has just gone. So therefore, the population of black flies would increase. Give a reason for your answer, because there's no predators for them. They are no longer being eaten. Table 2 shows the estimated biomass of the organisms in the garden. What conclusion can be made about the biomass in food chains? Well, we've got our biomass and this is the order of our food chain as we went down. Okay, and what's happening to the amount of biomass? Well, it is decreasing. Okay, and it's decreasing by it's halving roughly each time. Okay, so we could say, for just quite simply, as you go down, it's only a 1 mark question, so don't overthink it. As you go down or go up a food chain, the biomass decreases. And that would be enough to get you the 1 mark. Question 5.5, you then need to complete the bar graph, okay, and we need to label the y-axis and plot the data from table 2. So we've been given that bean plants go up to 225. So we know that each of these little squares is then worth 5. Black flies is 115, so we can go up to 100, and it would be up 3 squares because it's 115. Okay, and then trying to make sure that we draw the bars. Obviously you guys would do it with a ruler. Then spiders is 65, so again it would be 3 little squares above the 50. And then the blackbirds is only 10, so it would be 2 squares up from the bottom. Okay, and that is it plotted. We then need to plot or label the y-axis, and when you do this you always want to make sure you use the same label that is on your table. So it's got organism at the bottom and it's got biomass in grams at the side. So make sure you use the same label from the table that they give you. Question 5.6 then. Explain why a garden is not a stable community. Well, if you had a stable community, that means the population of the different animals would be staying the same. Okay, so stable, the amount of animals or organisms would be the same. As we've just seen in the garden, the spiders got wiped out and black flies are increasing. So therefore, the amount of animals in the garden is not staying the same. So we could say, why is it not a stable community? Well, you've got organisms are increasing or decreasing in number. Okay, so the population size is not consistent or is not constant. You could say, on the first point, adding a bit more detail, they are increasing or decreasing in number because they might be being added or removed from the garden. Question 6 then. Some students investigated the effect of drinking caffeine on reaction time. They used a drink containing 32.25 milligrams of caffeine per 100 centimetre cubed. This is the method they use. They divide the students into 4 groups, A, B, C and D. They measured and recorded the reaction time of each student using the ruler drop test. The students in groups, this is what they had. So group A just had water. Group B had 200 centimetre cubed of the caffeine drink, C 400 centimetre cubed and D 600 centimetre cubed. So the amount they had was going up in B, C and D. They repeated step 2 after 15 minutes. Describe how to do the ruler drop test in 3 bullet points. So normally, hopefully this is a practical that you would have done in your class, but what you do is point number 1, you work in pairs and you hold a ruler above the open hand of your partner. So one person has got their arm over the desk with their hand open, the other person holds the ruler above it. Normally you make sure that the 0 point on your ruler is between their fingers. Okay, you would then drop the ruler at a random time. You don't tell them when when it is going to do it, and the other person will catch it. And then finally, you record the distance on the ruler. And then the last point that you could say is that you could use a conversion table to convert the distance into a time. So there are plenty of conversion tables out there that you can convert the distance in centimetres to a time using a conversion table. Question 6.2. Table 3 shows the mass of caffeine taken in by each student. So you've got A, B, C, and then D is X. Calculate value X. Well, we've been told, if we go back to the question, that it's going up by 200 centimetre cubed each time. Okay, so therefore you're adding the same amount of caffeine as you go up the group. So it starts off with 64.5 with the first bit of caffeine, and then that's doubled, okay, to get it to 129. So you've added 64.5. So you would do the same thing again, add 64.5, which gives you an answer of 193.5. So 129 plus 64.5 equals 193.5. Question 6.3. Why did Group A drink water instead of a caffeine drink? Well, we always try to leave in an experiment a group or a person that doesn't have anything of what we are testing. And we call that a control group. So it is a control group, and the whole point of a control group is to see the difference with no impact of the drug. So it's effectively giving us a baseline measurement. Table 4 was used to convert the results of a ruler drop test into reaction times. Estimate the reaction time for a student who recorded the distance of 23 centimetres. So we can see, looking at the table, okay, it's going up in 2 centimetres. So therefore 23 centimetres is going to be between 22 and 24. So therefore we would estimate the reaction time would be halfway between 0.212 and 0.221. So what we would then do is try and find the midpoint of it. So the easiest thing to do is take them away from each other, 0.221 minus 0.212, and then whatever that value is, divide it by 2 to find the midpoint. So it should be roughly 0.217 seconds. Okay, so we're just finding a number that's halfway between 0.212 and 0.221. The students calculated the decrease in their reaction time after the drink compared with before the drink. So they're comparing their reaction time after compared with before. Figure 6 shows the results for each student in groups A, B, C and D. Describe the effect of the mass of caffeine taken on the decrease of reaction time. Okay, so therefore we know that group D had the most caffeine, and A had none, and B had a little. So therefore, what pattern can we roughly see? It is a weak correlation, but we can still see a general pattern, which is as the mass of caffeine increases, the reaction time decreases. So therefore, because it's going up, and remembering your units, it's a decrease in reaction time, so therefore they're getting faster as they do it. Question 6.6. For 3 students, the decrease in the reaction time was negative. Give a reason why their value was negative. Well, if it's at negative of a decrease, it actually means their reaction time got longer. Okay, so their time got bigger or longer, or the reaction time was slower. What is the range of results for Group C? So we have to go back to our graph now and we're looking at the range. The range is the difference between the highest and lowest. Okay, so if we look at our highest value, we can see it is at 0.18 and the lowest value is 0.1010. Okay, so we would then subtract 0.18 minus 0.01 to give us 0.07, or sorry, 0.17. Question 6.8. Suggest 2 control variables that should have been changed in this or controlled in this investigation. So we know that we are changing the amount of caffeine, but what else should we keep the same about this practical? Well, there are loads of things that you could put on this, so I'm just going to read out a quick list. The people in the experiment should have roughly the same age, potentially be the same sex or have the same gender. They could have the same height, weight or BMI. They must all have had no caffeine or medication earlier that day. They should have had equal amounts of sleep. They should have had equal amount of practice during the ruler drop test. They should have had the same starting point on the ruler, so that's making sure it's dropped from the 0 mark. You should have been using the same hand, and they should have had the same number of students in each group. So any of them would have got you 2 marks. Question 6.9. Explain why the ruler drop test does not involve a reflex action. So remember, a reflex action is one that we automatically do because we don't think about doing it. Okay, so like breathing, we are constantly breathing, our autonomic nervous system is controlling it. Okay, also blinking is an automatic reflex response, we don't think about doing it. However, with the ruler drop test, we are thinking and deciding when we clench our fist to catch the ruler. So therefore we could say the ruler drop test is not automatic because our brain is thinking. It is a conscious action. Okay, so 1 mark for saying it's automatic, 1 mark for saying, it's not automatic, and then 1 mark for saying that it's because it's a conscious action, we are thinking about doing it. Okay, so last question on this paper then. There has been a rapid increase in the percentage of carbon dioxide in the atmosphere since 1960. Carbon dioxide is a methane gas, a greenhouse gas that contributes to global warming. Name one other greenhouse gas. The greenhouse gas, the other key one is methane. Okay, methane comes from agriculture and it's a much more stronger greenhouse gas than carbon dioxide. You can also say water vapour in the atmosphere does also absorb heat as well. 7.2. Global warming causes climate change. You have 2 effects of climate change. So you can say the obvious ones, it hasn't limited you on anything here. So you can say ice caps melting, sea level rise. You could say extreme weather. And you could also say loss of biodiversity or destruction of habitats, etc. So there's loads of different effects of climate change. You just need to make sure they're precise enough. You can't just say things like forest fires or Earth getting warmer. That isn't specific enough. Question 7.3. Plants take in carbon dioxide from the atmosphere. Figure 7 shows part of the carbon cycle. And you've got the diagram here. Emphasis, the key part of this question says part of the carbon cycle. Describe how carbon from the atmosphere is cycled through living organisms. So it's effectively, you've got part of the carbon cycle and you've got to describe how carbon moves around it. Now, when you talk about the carbon cycle, you want to be including these 4 key processes: photosynthesis, respiration, combustion, and decomposition. So, starting off then, carbon dioxide moves into the green plants for photosynthesis. So you can start it off, point 1, CO2 moves into plants for photosynthesis. And the whole point of photosynthesis is to make glucose. Point number 2, so this is this arrow ticked off, so I would methodically tick off these arrows as you're doing it. For green plants, this is the part that most people actually miss off. For green plants, release CO2 when they respire. So green plants actually put CO2 back into the atmosphere when they respire as well. It's just less compared to the CO2 that they take in. Point number 3, animals can eat the green plants, which is represented here by the green plants, the energy from the plants going into the animals. Then what happens to all plants and animals eventually? All animals and plants eventually die. And then when that happens, decomposers, which are bacteria, break the organisms down and release CO2. So when we die, our bodies will get broken down by bacteria, and then that carbon dioxide or carbon in our body will release back into the environment as CO2. So that's representing the death in this arrow and then the decomposition in that arrow. And then finally, you can also say that animals respire in this arrow, which therefore again puts the CO2 back into the atmosphere. So point 5, animals, when alive, also respire, releasing CO2 to the atmosphere. Now that is everything they've put into the diagram. And if you just said all of those points, you'd at least get 4 or 5 marks. But to get the final couple of marks, you have to include the other part of the carbon cycle they haven't included, which is what can happen to the dead microorganisms. Yes, they get broken down, but they can, if they're trapped under sediment, their remains can be converted over millions of years to fossil fuels. We are now digging up these fossil fuels and we are combusting them. Combusting, which is the scientific name for burning. We are then burning these fossil fuels, which again is increasing the amount of CO2 in the atmosphere. And that is what is contributing towards climate change. So if you go through these key points that are down here and you add in the missing part of the carbon cycle, which is about the formation of fossil fuels, that would then get you the 6 marks. Okay, so that is the end of this biology paper 2 paper. I hope it was useful. And again, as I said at the beginning, there are links to the blank paper and Markscheme on the system and the description. And then also there's question by question revision videos for you to go back if you need to revise the content. Thank you very much.